Pilot, we will wait until your confirmation of thrust before releasing the vehicle.
Roger. Pilot has control.
Pull the pin. Pilot, lift line is clear. Let's go ahead and lock in auto head in versus ship and push center line.
Copy that. Pushing center line.
It's a good heading there, pilot, if you want to push forward.
Copy that. Pushing forward.
Okay, pilot off stick except for auto head.
Off stick except for auto head.
Alright, Dick, let's go ahead and launch Sirius. Pilot, please provide some aft way to reduce the tension in the tether as Sirius comes up and over the side.
Roger. Backing.
That's looking good, pilot. Let's maintain.
Copy.
You can ease slightly, pilot.
Copy.
and pilot off stick.
Off stick except for auto heading.
Winch, 0 reps.
Rep 0.
Pilot, 0 turns.
Turn zeroed.
Okay, tucker lines are clear. Pilot, are you ready to descend down to 50 meters?
Pilot's ready.
Standby 1. Alright, let's descend down to 50 meters, 15 meters per minute. Calling out every 10. Descending.
Going down. Yes, please. Video. Thank you. D2 crossing 10 meters.
One 0 meters, wire out.
D2 crossing 20 meters.
20 meters, wire out.
Good tracking, both vehicles.
Copy that, Nav.
D2 crossing 30 meters.
30 meters, wire out.
D2 crossing 40 meters.
40 meters, wire out.
D2 holding at 50 meters.
Good copy, pilot. Vehicles holding 50 meters. Winch, dive super.
Go dive soup.
Could we pass control to the control station at this time? Control room.
Roger that. Control room has it.
Co-pilot confirms control.
Good copy, co-pilot. Pilot, dive super.
Go ahead, dive super.
The control room is ready to continue with the dive. Let's continue descent.
Control room's ready.
Good copy. Nav, dive super.
Go ahead.
How's our approach to bottom?
We're going the right way.
Copy. We'll go ahead and pass control up to the bridge as is, and then I'll pass comms to you now that we're coming to hold station when you're ready.
Okay. Understood.
Nav, dive super.
Go ahead.
So you're aware we are currently passing control up to the bridge. They're going to maintain what we got. Please watch streaming as we do so.
Understood. Watching, streaming.
Nav, dive super.
Go ahead.
Bridge has good control of the ship. I am ready to pass communications over to you so you can work with them to come to waypoint 1.
Roger. Nav has comms. Nice launch, dive suit.
Nav has comms. Nice launch.
Go ahead, ROVF. Doing great. Nice day. How are you?
Pretty good.
Excellent.
If you guys want to work and get into auto position, and then let me know, and then I'll call moving.
Copy that. We'll get into autopause. Harvard Ave, bridge. We're full autopause. Copy that. Standing by. Copy.
Let's do 200 meters, bearing 211 degrees, speed decimal 2.
Alright, I copy. Move 200 range, bearing at 211 degrees, speed 0.2 knot.
Good copy, Bridge. But I don't know what you'll be able to do.
Copy that. Yeah, we'll start at point 2 and we'll see what demand's looking like. You want me to tell you what we're at or just do it?
You can just do it.
Copy.
Nav winch.
Hey, winch.
Hey, everything looks good down here. No obstructions, no leaks. I have permission to go up comms.
Permission granted. Nice job down there.
Copy. Likewise. Thanks.
This is Okeanos video engineer Roland Bryan making a general announcement to all those on the ship and the shore. This is a reminder that all audio associated with this dive will be recorded with our archive video, as well as being streamed ashore via the satellite and the internet. Keep that in mind as we proceed with the dive today, and enjoy the dive. Thank you.
Nav winch.
How you doing, winch?
Hey, before I go off comms, I just want to let you know, it looks like there's some vibrations on the wire. I don't know if you guys are seeing that up there. Might be due to the currents. Just wanted to...
We're doing half knot.
The moment, and we're feeling pretty confident that we've resolved the issue. So being that this is a shorter, shallower dive as far as the Okeanos and Deep Discover are concerned, we still think we can get a meaningful full dive in. So we've moved about 55 miles as the crow flies from our last position on in Ernest Strait. I'm getting my straits confused. So in Ernest Strait, and we've moved over to Behm Canal. And we found a nice site, which should be another vertical-ish feature, not quite as vertical as what we were seeing yesterday. But I think it'll serve as a good comparison to what we were seeing yesterday in a slightly different area, and a place where it's a little bit closer to the entrance of Dixon Entrance. So it'll be interesting to see any differences there. And with that, I will pass it over to my science leads to introduce themselves.
Thanks, Sam. My name is Jamie Conrad. Good morning, everybody. I am the geology lead on this expedition. I'm a geologist with the U.S. Geological Survey. I work at the Pacific Coastal and Marine Science Center in Santa Cruz, California. We're looking forward to today's dive. As Sam mentioned, we're in Behm Canal, and like yesterday, we're off of the oceanic plate and the Pacific plate and the seamounts, and we're actually onto the North American continent. We're about 100 miles west of the continental margin which is defined by the Queen Charlotte Fault here, which is a strike-slip fault, much like the San Andreas Fault of California, and somewhat different from the Aleutian subduction zones and Cascadia subduction zones where the Pacific Plate slides underneath the North American Plate. Today we'll, according to the geological mapping that's done onshore, we'll be looking if we see bedrock, and we actually fully expect to, based on what we saw yesterday, that consists of Cretaceous through Permian sedimentary and volcanic rocks, and that fairly broad time period, which is something like 150 million years or so, tells me, first of all, that the rocks were very hard to identify, and so they may be fairly well metamorphosed, and but we'll be looking at what we can see in them, and it's possible we may even be able to pass through a contact. It looks like one of the contacts that's mapped on shore is underneath our dive track, so if we make it all the way to the end, we might be able to pick that up. So, and with that, I'll pass that on to our biology lead.
Thanks, Jamie. Good afternoon, everyone. My name is Merlin Best. I'm the biology lead on this expedition. I'm a biologist with Fisheries and Oceans Canada. I work out of the Institute of Ocean Sciences in Sydney, BC on Vancouver Island for the Integrated Conservation and Monitoring Program. Am I interested in the dive today? Among other things, I'm hoping for corals. So this general area has been identified as one of potentially high coral abundance, particularly for the red tree coral, the Pacific red tree coral, Primnoa pacifica, which is what I would really love to see. A species distribution model by Chris Rupert and others from 2017 highlighted an area nearby, so in this general region, but it doesn't reach further inland to these canals that we're exploring. So we don't really know what to expect here. I found a paper from 1981 looking at bycatch and anecdotal evidence from fishers and scientists, and it mentions Behm Canal specifically as having frequent reports of Primnoa, so that larger structure-forming coral. But there aren't any exact locations, so we could easily miss them on this, exact dive site that we picked. For example, yesterday we didn't see a lot of Primnoa. We saw some, but a really high abundance of a different coral, black corals, the Bathypathes alaskensis, which was very cool to see, that local high abundance. And I'd say that this is the fun part, waiting to see what appears out of the depths when we hit the bottom, and explore this line that we drew on a map that we just came up with overnight.
Yeah, so if you're looking at Stream 3 right now, you should see the bathymetry in the region that we collected last night and kind of how we selected this site. An interesting thing about this, it's colored by slope right now, so you can tell where the areas of high relief are. And this is with no vertical exaggeration. So this is a scale model, true to life, of what you'd expect to see down there, kind of expecting what D2 will see when they fall, when it reaches bottom. So it's a very dramatic landscape. So it's an exciting place to dive and should provide an interesting place for things to land and grow and live, hopefully.
And so what we're looking at is actually a fairly young landform. These fjords and sounds, and I guess this one is called a canal, but they're all the same thing out here. Were all filled with glaciers during the last ice age, and the glaciers were last here. They began to melt away and were gone by about 15,000 years ago. So everything, all these landforms were carved at about that time, or by then, and So that's what we're looking at. That's fairly typical for this whole mass of islands in this archipelago known as the Alexander Archipelago. I was going to mention that we were in the archipelago. It's a group of more than 1,100 islands that make up part of Southeast Alaska. But I looked closer at the map and I realized that we are just barely in the archipelago. In fact, to our east is actually mainland. And so we are right on the boundary of the archipelago. To our west, where we're of our dive site, is an island, one of the southernmost islands of the archipelago. We're located about 20 miles southeast of Ketchikan. And again, the steep sides of this canal that Sam demonstrated are a typical feature you see in glacially curved areas. This is kind of a little bit of a mini Yosemite. This looks pretty dramatic. We're looking at elevations, though, of a couple hundred meters upwards, but makes pretty good cliffs in places.
I was thinking about that, how we're quite a bit closer to my normal study sites. We're just north of Haida Gwaii. So in some ways, geographically, a lot closer than we have been, but in other ways, this is very different from the places that I normally look at in the offshore region off of BC, particularly seamounts in protected areas or potentially protected areas. Looking in these really constrained, these like canals has been very cool. We also spent a bit more time than usual in the water column yesterday, in these seats at least, and I got to brush up on some of my mesopelagic identifications. So all of the little critters that you're seeing swimming around in the screen in front of us. Noticed this yesterday too, just a really thick batch of marine snow falling the entire time. So all that dead organic matter and debris kind of in these big flakes that will eventually fall down to the sea floor and feed the ecosystems there. Getting very familiar with these little squiggly lines that you're seeing are probably Ketignaths, or they're also called arrowworms. They're a pelagic worm that lives in the water column. Sometimes if you get a close-up look at one, you can see these really vicious-looking little teeth on the front.
Oh, my.
Yeah.
How do I see
these.
okay.
But we won't. There's two modes. This mode won't do water column velocity. It'll only do bottom up.
Got it.
But which is
I was
obviously
wondering-
upset about something right now.
I was wondering if we would be able to see like how fast the water's flowing by.
Could you bring a pose cam real quick?
Yeah. Ox 2? Or, yeah.
Yeah. No.
Actually green. Or blue, I'm not sure. Looks green, but... Doing a heading change, gentlemen. You're going to swing a little bit. Be advised.
Copy.
I can do a ship move to trim it out. Looks like layback is kind of settling out. See that?
Yeah.
So I might make a move to account for that layback.
Okay.
If that's alright. Wait, what's the heading change? Yeah, you can just do your heading change now. We'll get you comfortable first. Understood.
I wonder if those lines are interfering. I remember we talked about using acoustically transparent material there.
They're bringing the head to port. After that, I will make a ship move.
Do either of you know if they're looking into a more permanent fixture instead of the lines?
I don't know.
There was talk at one point of something a little more constructed and machined to serve the same purpose, but
When it initially came up, we were talking about using acoustically transparent material. I think RDI said like branded plexiglass was one, which was kind of surprising to me.
huh.
Working on HD2, HD1 is clear.
Meters 164.
All right, did we also get R3?
Art, did
Art,
you
get his picture? Yep, Art's all set.
a little more rotate to get the left colors? That's good.
Because that looks a lot like 0.3 meters to me. Or pretty darn close.
Yeah, could be. It's ripping down here today. Look at this.
I'm actually a little bit concerned that there might be too much current.
I'm 90 degrees down in the camera, so it's just ripping.
I know. Concerned he's not going to have enough axial thrust to get back underneath you. Alright, what's 62 plus 180? Hey, Bridge. Okay, so you ready for another move? Alright, let's do 30 meters, 30 meters, bearing 164 degrees, speed decimal 2. Good copy, Bridge. I'm going to move you guys away from the steep section. That's what I'm working on.
Roger.
Okay.
Yeah, I'm not pushing out at all, and it's been like taut the whole time.
I suspect once we get there that we're going to have trouble getting D2 under steers. Especially if you turn broad. D2 turns broad to it. Beam to it. Right?
Yeah. Which D2 will be.
My only grasp for water speed past Sirius is looking at that camera. It is ripping. We know the field of view. We could.
Yeah, we're holding position, right?
Yeah.
You can see how fast he's going past the rails.
You're pretty much holding position, yeah. It's Sirius is moving a little bit, but to starboard. See this? I know. Now I'm moving it south.
This camera just doesn't have the handles I wish it did.
Bridge over now. You can do 0.3 if you want. Thanks.
Well, I'm just trying to do some fine tweaking on this camera to improve it. If you guys need me to wrap it up, let me know. But otherwise, I'm going to try to just do a better job on this camera.
Yeah, that's real. Let's see. 400 would be 500. I got you at 550.
Copy video.
D2 looks kind of big, doesn't he? It's a normal scale we do.
So we've got a move underway right now, right, Nav?
Yeah.
Maybe when that move's done, we'll wrap up the white balance and go down.
All right. I did a 30-meter move, and they're halfway through it at 0.3, so 15 meters, 0.3, 2 minutes. Yeah, 2 minutes rolling.
Copy.
Sirius has moved.
How much is our tether stretched?
50 meters.
4.3.
Whoa.
According to acoustics.
Ship length was about what it was yesterday, too.
It just, in this last move, it looked like it stretched even more. It's starting to show. It just had frozen for a second.
Yeah, you're going to have to use your axials in your last today, Andy, like you mean it.
Full power.
Yeah.
Might be better once we get deeper.
Do you want to start going down a little bit more?
Sure.
We're basically done with that move.
What do you want to aim for?
So, like, for when we stop for the setup?
Just
go ahead,
what do you
Rich.
want to go to right now,
Move
or
quickly,
set
thank
up,
you.
or?
We're going to proceed down now.
Okay. I'd say let's go down until Sirius is, like, at 30.
Okay. And you're still pretty close to that wall.
Okay.
But we can start heading down.
You're moving away from it. So, let's see. I'll zoom in on you guys here.
I mean, that's what I was thinking for the setup, but
Raj?
we'll watch out for the altitudes.
Coming down 30 meters a minute.
The video is clear.
Thank you, video. Thank you, co-pilot.
Can we follow you around?
Aircraft secure.
It's your estimated 150 off bottom. Might start seeing it come in.
You have your camera tilted down on the main?
What was that?
You're just checking to see if the main camera's tilted full down?
It's not. I'd believe that. 145 off bottom.
Yeah. And we're, as far as moves go, we're wrapped up.
Yeah, you're stopped. As you're paying out on the winch, your layback is increasing a little bit. So you're, does that make sense?
Yep.
See that?
Looks like your 100-meter theory is accurate today.
Okay.
So you're still over by that nose at 550.
Yeah, Les, I imagine once we start setting up, we'll know. But yeah.
Okay, approximately 100 meters off bottom. Gonna slow down a bit. Delta's growing.
Watch your RV sonar.
I'm at 70 meters altitude.
Copy.
I think I see something in blue on your port side
Yep,
there.
you got it port
60,
side where we would expect it.
50 meters out.
So I'm in deeper water than you, I think.
No, I'm deeper.
Yeah, that's what I meant.
There we go. My altitude just jumped back up to 98.
Yeah, D2 should be in 560 meters, and Sirius
I believe this altitude now, 90,
575.
90 meters altitude.
Okay, and that matches up with the DVL
Yep.
and the delta.
Those previous pings were incorrect.
As you pay out, layback increases 36.
Looks like we're about
a lot of
50
current force on the cable
meters
and Sirius.
from the bottom. Eyes peeled on the main camera feed. One of my favorite parts is when it
Oh,
comes
so
into
what
view
we'll
and we get
try
the
for is
first
I'll back
glimpse
up
of Behm Canal,
and
maybe
I turn
ever.
Port, correct?
What it looks
That's
like on the
direction
on the
that we want
seafloor.
for the tether.
I don't remember.
Typically, they want D2 to turn starboard.
Starboard, so I'd
So
be
you
going
could-
all the way around.
Well, you could turn port and have co-pilot turn port. You know what I mean? You could turn all the way around if you wanted to keep the tether in that situation. But a lot of turning today might not be what we want to do.
I think we're just going to have to accept that
Yep, I agree.
D2 turns left, I turn right.
And then
Okay.
for the record, I have from Sirius,
So I'm at 50 meters altitude.
Okay, so you can hold there then.
Okay, coming to a stop.
And I'm going to go down a bit more and then start backing up. And would you get your...
Auto head in.
Okay. I've got mine in. Auto depth engaged. C-bias secured. And would you point your Zeus all the way back?
It
Oh,
is.
you got it. Thank you. Okay, could you get me in Titan when you get a chance? Not sure how much progress, backward progress I'm making.
You're gonna have to, oh, you're making some ground, Andy.
Okay.
On high pack, you're making some ground. That's good.
So am I, apparently, though.
So yeah, you're taking out some of the layback. You're saying?
Right. And that's Chem 3.
Yeah, I don't have myself in, I'm off tail, but I'm not on.
So you're still on the digital. When you kind of come out of that screen at about 20,
Oh, that's my light pool, I see.
20,
Okay.
you'll come into the downward digital. I can't quite find you on Titan yet.
All right. I'm back in pretty much at 100% now.
You're making ground. You're making ground.
All right.
Look like you're going a little bit off to Sirius's port. You may want to
Okay.
put some port way in according to tracking. I don't know if that lines up with reality, though. Bobby can help
It
you
looks
with that.
like off the tail. I am as well. Okay, I'm going to put in some port.
So I'm going to bring my lights down.
Okay.
Might help you on the downward.
Okay.
See you in Titan. See you in Titan.
There you are. See, you're right below me.
All right. I'm going to go just full back now. This is 100% back. Looks like I'm just kind of holding.
Like you're making ground on high pack. You're making ground.
Delta 17, you're here now.
Oh, got it. Thank you.
I'm going to bring one light on, see if it helps us.
Yeah,
Okay.
you gotta port
really?
that. Yeah. Problem is once you port that, then you drift backward.
Center it up.
Lights, all lights are at full. Can't really.
It's because of the delta.
I'm trying to think if I can aim a light on the foam pack or something. Let's try uppers down.
Tether, tether, tether.
Oh, wow. Okay. All right.
Mini Zeus.
Yep, I see it. Thank you. So I'm going to go.
It's off the port side.
Yep. So I'm laterally to starboard. And I'm going to go down a little bit.
Good. Went up and up. Went up.
Okay. I'm going to go
Get
down
some more delta.
Yeah, I'm at 20.
I can't see all the.
And I'm going to back again.
You see your heading.
I think I lost some ground while I was doing that.
Bobby, you want to go inverse to his new heading?
I can. So, pilot, would you like that?
Sure.
Okay,
Yeah,
come
all
to
the way down
starboard.
the water column fed a really high
like there you are,
density of
bottom
this
left
marine
again.
snow.
Got it.
Starting to see
Thank
little
you.
shrimp.
Lights coming out.
I think little
Okay, I'm
there
going
you
to
go.
starboard over.
some ctenophores. Those
And I'm
bigger
gonna go down
blobs
a
that you
little
see
bit.
could be
Still
comb
coming to
jellies or
starboard.
ctenophores.
Okay, I'm inverse.
Another shrimp
Copy.
flying
Hey,
by.
you guys are heading to inverse, yeah.
Okay, I'm still lateraling over and keeping my backing to center up.
I think we're about 10 meters or so off from our
Just lost
target, so we'll see.
I'm going to go up a little bit.
Oh,
You got a port lat, not starboard lat, I think.
Quick visit by someone.
Yeah.
I was going the right direction.
Yeah, they're
I'm
fast.
Still backwards, but coming.
You're right below me. You can see yourself on that camera.
And I see the tether,
There
too.
you go. You can see a halo of
Okay.
light. It's not very good.
All right.
And of course, we're bringing
Video.
our own light with us all the way down.
Try.
Sometimes it even attracts some of these mesopelagic animals.
You have a halo here, left
Okay.
side.
All right. So if I...
You got a port lat.
Okay, I'll port lat.
We're still opposite, so port lat should bring you center. Let's bring lights all up. I've tried adjusting angles of the swing arms, and I'm not getting anything back. Here, fogs.
You center it up.
I don't know if that's going to give you anything. Ox was already at 100.
All right, so...
It's good you're centered up now.
All right, so...
I'm still all the way back on my camera.
Okay, so I'll back up some more.
Yep, delta is 22, looks good.
This is back at 100.
If you guys aren't comfortable, we can call it too. Seems like an enormous amount of current.
Yeah.
Tether through the ox light, I can't tell where, if it's your end or what's happening. I just saw it pass through.
Okay.
Ox light, so be on the
Well,
lookout.
what do you think, co-pilot? I'm kind of set up. I can turn around and we can get in a setup and see.
I can now see the front of your vehicle is facing Sirius.
Yeah.
I got uphill bearing 330, gentlemen.
330. I'm off stick right now.
So we're just pausing a little more than 10 meters off the seafloor to do a little bit of repositioning.
Drive under me.
And you see the fog lights have really lit up all of that
north. That's partially why
marine
we
snow.
got it on our port side.
All right, so I
Another
can go
little
starboard.
arrow worm or a Keating nav.
That would be, I would be doing the correct tether.
I think that's probably what I would try.
All right.
I would.
You at 90 now?
No, I'm still all the way back. So you need to back until you're
Okay.
center, because you're going to start drifting on the turn.
All right, you can go to 90 now. Should be able to still keep me in.
Okay. It's 94. Dive soup's here, so let's hold on anything.
Okay.
Just maintain position.
Maintaining.
So again, it looks like we're just pausing the dive to do a little bit of repositioning. We'll be back soon with an update.
So I'm going to turn to starboard then, co-pilot, and I'll drive under you.
Yep.
You're at 94.
94.
Would you go to 90 just so...
Okay. I mean, you're going to start to drift as you turn around. You're going to lose all your... I think let's focus less on what that angle is and just try to make the turn.
All right, here, I'm going to get a little bit ahead.
You can turn faster manually.
Yeah, that's what I'm going to do.
Send it.
All right,
I'll
ready?
watch
I'm going to turn.
I see the tether. Looks good, good, good. It's going around. It's going around.
Yep, clear tether.
There it is.
Okay, you're there.
Okay.
Too much, too much, too much.
Back left, yeah. That's good. 3-3.
Hold,
good.
hold.
I got it. Hold on.
300.
All right, there we are.
Yep. Center up again.
Lateral starboard. Push forward.
Laterally to starboard.
You gotta push ahead hard.
I gotta push back, don't I?
No, push forward.
Forward. All right,
hard,
pushing
forward.
forward.
Hard. And starboard. Got you in tracking here. Looks like you got a lateral
Forward and
starboard.
starboard. All right, I got it.
Yeah. There you go. Full warp. Looks like
light speed.
Star Wars.
It's because the swing arms are also all messed
Turn
up.
fogs off. It's not helping for long range. Blue View's got it. 30 meters down.
So this is full starboard and pretty close full forward. Yeah. All right, so you're going to change your heading, Copilot? What was it, 330?
Yep, 330s uphill. Now, once you do that, you're going to kind of go beam to it. And your lats aren't very strong. So don't blow off.
Bringing my heading around slow.
Okay, I'm bringing mine around.
So you're going to have to, like, port lat real hard pretty soon. You're going to blow off.
$330.
330, aye.
The boat, let's come down.
High peck as you're moving starboard with the current. You're going to have to port lat, port lat, port lat, port lat. Yeah.
All right, I'm going down.
Okay, coming down slow. Showing 22 altitude.
Okay, I've got 25.
I must be reflecting off you.
Blue's got it. 25 outing closing.
All right, so that's...
Blue's got it.
Yeah, I have 46 now.
DVL.
Paying out about 15.
Blue View, 20 meters out. Looking good. You're normal to it. It's looking good.
Bring my camera up. That's 75.
Okay, 15.
You're right in the middle of the camera. You're looking good.
Delta 20.
Okay, I'm going down.
Blue View's got it. 18 out.
15 meters altitude.
You can range in then a little bit now.
Roger, range in Blue View.
18 meters delta.
15 meters.
12 off bottom.
Okay, I'm at 12 off bottom as
Yep.
well.
No, I'm 20 plus. I was reading your altitude. You're 10 off bottom.
Basically 100% of your lats, huh?
Yep.
Yeah.
I'm hoping when we get down to the bottom, it'll be lower. We've seen that before.
You have bottom.
Okay, I've got visual.
Winch is all stopped for now.
Copy. Okay, I'm just going to...
All right, we're on bottom.
I'll stick briefly.
Look at this. So we were just having a discussion about the turbidity here. There's a lot of material in the water column, but can already make out a couple of features on the floor. I think that's a sponge
Hey,
of
just so
some
you know,
kind.
we're on bottom. There's quite a bit of current down here. Let's set bailout.
And just to give you an idea of why that's an issue, where if you look at camera 2, you can see the view of D2 from Sirius, which is tethered above it.
Are you comfortable coming down to like a 10 delta?
Yeah, I'll come down a little bit more.
And you can see it's a little difficult to see D2, and we like to keep an eye on it from Sirius so we know what's coming. And
that's my concern there.
It helps a lot, and so we're spending a little time getting oriented to make sure we can keep track of D2 through all this turbidity.
Just get a little bit better view of the vehicle.
Roger.
Something interesting, giving a bit of texture to the sediment down here, maybe shells like what we were seeing yesterday.
Right,
Some kind of
eyes
organic debris. You know, yesterday, aside from the black corals, which I was super interested in, pretty much the whole, vertical wall of that dive, everything was covered in these brachiopods.
I think this is doable.
Don't have an idea of what species they might be. We were able to sample one because they're incredibly abundant here. But these, there's some different kinds of shells. Looking at the debris that we're seeing right now, which gives us an indication of what might be further up the wall.
It is, and we saw that yesterday at the bottom of that wall. We saw tons of shells that were almost exclusively brachiopods shells. And here, I'm not seeing any. I'm seeing something else, but I have no idea what. There's one rock over there on the right, but...
Where?
Bottom right corner. Yeah.
There? Okay. All right. I thought I might be seeing some really big, kind of looked like a really big barnacle almost, and then some of these other ones I think are a larger bivalve of some kind that's been crushed. Oh, there's a nice rockfish up ahead. So once we get situated a little bit, we might be able to get a zoom on these and take a look and hopefully find some living that we can get some close-ups of to kind of see what species are making up the bulk of the ecosystem we're finding here. Ooh, thanks for that. Sean Rooney is joining us in the science chat. If you don't know about that, it's our chat room full of our shore-based scientists. A bit fewer than normal today, being a Sunday on a long weekend, so I really appreciate the folks that have been able to join. So we have a tentative ID. The anemone could be Cribrinopsis fernaldi.
You want to scan up Blueview, Bobby?
I could I'd definitely use help with anemone identification. I have Metridium and Lipodema, and that's almost the extent of it.
Watch leads, Nav.
Watch leads here.
Just want to give you an update. We're on the bottom. There's a tremendous amount of current here, so we're using maybe 80-70% of our thrust just to stay here. So we're going to proceed, but it might be kind of slow.
All right.
Sounds good.
We
Thanks.
might have to call a dive if there's too much current or something, but
oh boy,
we'll see what we can do.
Yeah, and at some point when we're ready, let's we might as well grab a water sample while we're down here.
I'll let the co-pilot know.
Whew. Good call.
Probably 330, Andy. Yeah, the move will be 330. You guys ready for a move, or do you want to wait a second?
Well, before we move, we probably should explore this section.
Let me range you out, Bobby, or are you good? Are you blue viewing?
I saw you moving the camera and stuff. I thought you were driving it.
No, I just got it up to see out a little bit.
No, why don't you drive it? I have enough going on.
Looks pretty gentle for 40 meters. See that? Right at the 40-meter ring, you get those hards. It looks soft and supple.
It's definitely
you guys good?
Not ideal.
You guys good for 15 maybe, just to get us going here?
Let's do it.
All right. 330 degrees.
We'll probably leave the swing arms in.
Bridge, this is RVNav.
Yeah, after seeing that
Yeah, I'd
tether
like to
come
move
in,
if you're ready.
I don't want swing arms out today.
Let's do 15 meters. That's 15 meters, bearing 330 degrees, speed decimal 2. Good copy, bridge. Looks like a gentle slope, Andy, and then it gets hard at 45 meters out, so I'll tickle you in.
Okay. Did we fire that Niskin?
We did not.
I think that's actually our first Primnoa sighting.
And whenever you guys are ready for a Niskin, we'll be fine.
We're doing that right now.
Roger, watch lead. I just turned on hydraulics. Got the thumbs up from back row, and Nav has given me the thumbs up. So here we go for the Niskin.
So we're taking a moment to collect a water sample that we're going to be processing for eDNA analysis. So that's the study of eDNA or environmental DNA.
3, 2.
There are folks that are using that information that is held in the water of animals that may have passed through the sample that we've taken in the past. They leave a little bit of their DNA behind, and we can amplify that and separate it out into different groups, and that can tell us a little bit more about the kinds of animals that at least pass through this water and live in this area.
And so that was a good capture. I have sample ID EX2306-D10-01W, Niskin Bottle 1.
Yes.
All right, I'll move forward.
RV, could we get a zoom on the anemone?
I'm sorry, watch lead.
No, that's okay. No, you just let me know.
Ship's getting right above me. I gotta move forward a little bit.
I was just given some very cool anemone facts about some of the larger pink anemones that we're seeing. They brood their young internally, which is wild to picture. A common symbiont of these anemones is the candy-striped shrimp. We've also observed the scaled crab, placitron, in the genus placitron, sheltering under the anemones. So that's very cool. Even those kind of solitary large anemones providing habitat for other organisms.
Good.
I think I'm seeing a solaster of some kind just passed over, and a little corallomorph anemone also below. Also giving us a great opportunity to get a look at some of this sediment. It looks like it's all broken up shells, different kinds. I think I see a gastropod shell there. Wondering if some of these tubular-shaped ones.
We normally leave it at 50, though, don't we?
You know, hard to know until we see something living, but.
That's what I remember.
Yeah, there's definitely a lot of variety, a little bit different in composition from what we were seeing yesterday. Not a whole lot of opportunity for a geologist, is it?
Shell hash.
Mm-hmm.
That move's complete. I'm not sure how long it's going to take you to feel it, but I think this, oh, here we go.
I think it's already gone.
I can see it.
Because I was at 45, and then all of a sudden I was at 72.
Just like a really young limestone, don't you think?
IPAC may be having a moment. Be advised.
Copy.
Right, we see these in the record. Coquina is one term for them.
Like a layer of...
a shell hash, a layer, thick, mostly grain-supported with micritic, I want to say ground mass, but that's not the word I'm looking for.
Coquina?
Matrix. Matrix.
Oh, okay.
Yeah.
Coquina.
Coquina.
Okay. Oh, very cool. I'd imagine you might see them in core samples sometimes. Does that ever happen?
We sure have.
Mm-hmm.
Yeah. They often mark the end of the core because they're not terribly easy to penetrate, but not always. You can find them if they're thin enough. If they're just a couple centimeters, the core will go through. And we see them in the rock record if you're in limestones in different places.
Apparently they had core samples from the area of dead Lophelia rubble. There's a couple of locations. I think one's in the Juan de Fuca Strait, actually, a big pile of the dead Lophelia. There are living colonies that we've found off of Vancouver Island, but I think that they had a core measurement because they had an estimate on how thick it was. I thought it was from a core. I could be wrong. I don't know how that would happen. They're very tough.
Well, it's possible they have some seismic reflection data, and they can correlate, say, the top of that layer if they hit it with a core site, and if it's too thick to penetrate, they might be able to nonetheless recognize it in seismic records.
You want another move, Bobby? You're going to wait a minute.
Yeah.
And something like this, I would imagine, would give a different kind of reading with a seismic, you call it a profiler?
Yes. Yeah. Oh, yeah, this would be a nice hard return, so it would be you'd see it as a nice bright reflection.
Uh-huh.
I'll move you 330 one more.
But again, the important thing is always, what's preserved.
Bridges are we down?
Well, it depends on who you ask.
Well, for looking at things in the record. And so the ultimate fate of this, as spectacular and thick as it looks, is the next ice age is probably going to wash it away and carry it off. So
right.
Is our goal only to get to the hard structure?
To the extent that you can preserve this channel in the rock record is pretty tough. Eventually things are going to get planed off with erosion or uplifted and eroded. So uplift will cause this to sort of flush off, and erosion is going to carry all this out to sea.
Right.
So
I'm going to move Sirius towards you.
I see on the chat Arvind is asking us if anyone else is noticing the hermit crabs, and I'll have to say I'm not. So I'll take a guess. Is this another one of our sculpins?
Ooh, that would also be my guess. We had a tentative idea of a dark fin sculpin on our dive yesterday. Let's see.
But now my eyes are out for the hermit crabs.
Okay, yeah, there's a couple of people in our science chat that have seen them. So I've also noticed a lot more like woody debris. I think I've seen leaves and maybe some sticks, which isn't surprising considering what we're seeing outside. Look at these, I think they're some of the brachiopod shells rolling in the current.
And these corals, no, anemone, whipping in the breeze.
Yeah. So we were told that this is the time of day right now when the tidal currents are at their strongest. So we're hoping that they calm down a little bit as we proceed with the dive, make things a little bit easier to navigate. And yeah, so these large pinkish anemones could be Cribronopsis fernaldi. And we can keep an eye out for their symbiotic shrimp. So I imagine the shrimp gets a little bit of extra protection hanging out in and around some of these anemones and maybe a little bit of access to food. Oh, I think we saw some of these yesterday, those little yellow objects on the anemone tentacles. But it's hard to tell from back here.
Now, what are these things behind him? That's part of his foot?
Oh, there's a crustacean,
Is
a crab
there
behind
a crab?
him.
Okay.
Yeah, there's a crab just
Looks like
kind
a
of
leg.
under there hiding out. And I think that was also one observed, the scaled crab sheltering under the anemones. So I think that's probably what we're seeing right now. Possibly Placitron, a genus that I am not familiar with, so apologies for anyone who knows how to properly pronounce it. I think those are some of those little striped shrimp behind the crab as well. There we go. Sean has records of seeing more than a dozen shrimp on a single anemone. It's very interesting. I'd love to hear what the anemone gets out of it. A couple of different kinds, maybe a small pom-pom anemone, which we did see a couple of yesterday.
some signs that we're getting close to some rock, a few boulders, and I see something on the sonar from D2, looming just a few meters ahead.
Yeah, I think that was there's another one or two of these Liponema sea anemones. I think there's some seaweed down at the bottom, which is maybe the first seaweed that we've seen
Yeah, I think we should
on this trip.
focus on the dive right now, Levi.
At first I thought that was a sponge, but I think it's a retracted Metridium type anemone. There's a rock.
Especially as we're coming up to this ball. I'm in survey 1, Levi. I don't know if you can hear me or not.
So we're apparently nearing the top of this debris apron that lies at the foot of this steeper rocks. We're following up one of a number of several ridges that ascend up the side of this sound.
Very angular. And I can see already from here that they're just covered in encrusting fauna. I think a lot of that is sponge, different kinds of demosponges. But then I think, little specks of maybe those unidentified white things that we were seeing yesterday. And brachiopods again.
So we have a fair idea of what these rocks are from what has been mapped ashore. There's a succession here, and we can see the bedding or the fractures that follow the contacts. Those might be either depositional contacts, but more likely are fault contacts, given the ages of the rocks. These are considered to be Cretaceous through Permian, which is a fairly long geological time of something like 100 million years. And a range like that tells me that the rocks are first of all, not really recognized from other rocks, similar rocks, and they've not been easy to assign a date to, probably because they're metamorphosed and fossil evidence is gone in the case of the sedimentary rocks, or in the case of the volcanics. The original radiometric ages of the rocks have been reset by metamorphism. So if you were to date anything you found that was volcanic, you'd get an age of the an age of the metamorphism, and not the age of the original rock. But it looks like maybe we're not even going to see very much rock, since this is basically completely covered.
It does look that way. Yeah, just carpeted with these brachiopods. And I think I'm seeing some, lots of scattered smaller Primnoa pacifica, that coral that can get to really large tree-like, or tree-shaped sizes. Maybe like I think I've seen one approaching a meter, and provide a really rich, dense habitat for fish and other invertebrates and all kinds of stuff. So it's encouraging seeing some of the juveniles down here. There's it's like a dozen just in this one view. That's exciting. I think a couple of those are maybe something like the red-banded rockfish. But a rockfish species with alternating red and lighter colored bands.
It
I think I see another, that pink blob in the middle and up towards the top is a nudibranch, one of the genus Tritonia, probably, and we've seen them very interested in corals. I think they predate and they eat coral material. And a big celaster down to the bottom right. And yeah, this rock face kind of scattered with these really small colonies of the Pacific red tree coral, Primnova pacifica. We're pulling away from the wall for some adjustments and take another look at the water column, see if we can catch anything as it flies by in the current. So we've just been updated on the situation. The current conditions at the bottom, where we're close to that really sheer feature, that vertical wall, are a bit too, I think, treacherous. So what we're going to do is reposition to explore the top of this feature, which is something that we're both interested in seeing. So we'll just skip ahead a little bit and explore the top of this. And we're still within happy depth ranges that Primnoa and other corals like to be in. So I'm excited to see the top. So stand by. We'll be back once we do a little bit of repositioning.
bit on the vehicles. And then instead of landing at the bottom of the cliff, we're going to kind of try to land at the top where it's a little safer. Maybe we'll come down in this amount of time. It takes us to the game. So I'll call them in a little bit here.
degrees, speed decimal 2, please. Thank you.
wired the bottom. We're now at a depth of about 400 meters, a little bit up that ridge we were at the base of earlier.
Yeah, the rock is a little bit more clear. There's a little bit more for you to look at. I'm still seeing a couple of those big anemones. Liponema, I think. And some sea stars. I think a cookie star to the left, the Sarah Master, maybe. Oh, it's a little big as well, maybe something different. And yeah, quite a few more of the probably brachiopods, although they look a bit different from the ones that we were seeing yesterday. They were quite a bit larger and darker brown color. These ones are a bit smaller and white, so it'd be nice to get a closer look and see what we're dealing with.
I think it'll be a few minutes while they see if we're happy here with the currents, which still look to be blowing by pretty good rate.
Yes, they sure do. Can see all the small tubes and things projecting out of that rock are really whipping around. That crab seems to be doing just fine though.
Sounds good. Copy that. Do we want to get a water sample here?
Sure, let's do it. Can we get a Niskin? I think N2 is what we're looking at.
Sample ID EX2306-D10-02W, Niskin Bottle 2. Watch lead. I believe that was 575.
Okay, some of our, really good zoom can get a nice close look at this, all of the fauna that's growing on this rock. So I think, again, brachiopods, some of those sponges, and crustaceans. think that's that kind of pyramid-shaped gastropod that we saw yesterday, Calliostoma. And those kind of tube-shaped sponges are, they remind me of sponges in the genus Auletta, a edema sponge that kind of gets that tubular shape. But we were also seeing a lot of cool antler-shaped sponges the other day, so possibly something like that.
The rocks are a little hard to see, but we expect that they're the same thing down at the bottom of the ridge that we first saw briefly an hour or so ago. These are Mesozoic, metavolcanic, or metasedimentary rocks. It's hard to tell here much else about them other than we can see the fracturing and jointing. Those are the results of tectonic forces and the deformation these rocks have undergone. We don't know a lot about the origin of these rocks or their history very much, except for that they probably formed at some distance from here and were carried here and accreted up against the continent by plate motions along with the movement of the Pacific Plate.
RV, could I get a zoom on something? Oh, I think the coral that's kind of above the lasers, pinkish. There's a whitish sponge right above the lasers, and then to the left of that. Yeah, yes please. So I think probably could be a hydrocoral. Did see a couple yesterday. So I think I got a little bit of a look at it. I'm confident it's a hydrocoral. Most of those are in the genus Stylaster in this, that kind of tree-shaped morphology. So they're, kind of more distantly related to the other corals that we see than you might think they're in they're hydrozoans. So in a different group, and they generally stay pretty small, really little delicate branches, but sometimes you can see their little tentacles coming out of the the hard skeleton. Beautiful, and a good view of that little brachiopod as well. Some cereanthid anemones and little sponges. That's great. Do you think we could pan over to the sea star that looks like it's eating something? So it's up and to the left.
Chris Ma thinks those brachiopods might be in the genus Laequias. He's spelled it out phonetically for me in the chat.
Oh, that's always really handy.
That's very handy for me.
I think a few more hydrocorals, a really big orangey pink nudibranch, and then this sea star that I just had to go and take a closer look on. Looks like it's viciously attacking a coral, maybe. You can see the remnants of one just to the right of it there. I think this is a Ceramaster, so one of the cookie stars. And yeah, you can see the polyps of a coral, maybe something like, I would guess, a little, very small primnoa. Thanks for that. That's great, Arvi. I do wish we had time to flip it over and see exactly what it's eating, but I think we can take a pretty good guess. Oh wow, it's quite the shelf.
Maybe a little shear right there. Kind of a... Definitely some big blocky rocks with very prominent joints in several different directions.
Yeah, a lot more of these really large pom-pom anemones, the Liponema genus. And we're also getting really excited, some of the coral biologists on the chat are noticing juvenile primnoas, really small ones, which is always exciting to see some recruitment in this area. There's another one just coming up at the top. I think more small hydrocorals, a couple of boot sponges, I think, in the family Rosselidae.
Little bit of an overhang.
Jamie, I'll get your help. I'm showing you a picture here. There's a sea star on our wanted list.
Oh boy.
Yeah,
Okay.
we saw it yesterday, so you'll have to help me keep
All
an
right,
eye out
all
because
right.
there's a lot going on here.
It's got 5 arms. Got that.
Here, I'll keep it open over there. Hopefully we can, it'll be front of our minds. I think that it's one of those little, looks like a little snailfish to me. Has it died down a bit? Huh. Okay. It's a really dynamic area as far as the current goes. Also really beautiful. Seeing more of those darker brown brachiopods. I can't tell if they're just older individuals or a different species. Some of these little tiny crabs, I think we might have had a suggested ID on them earlier. Let's see if I can find it. Now we had the association of the scaled crab in the genus Placitron sheltering under anemones, so you might be seeing a couple of those, but these are different species, these Liponema pom-pom anemones. Seeing more of those little tube-shaped sponges, lots of encrusting sponge. And scattered hydrocorals, those really small pinkish branching organisms, or those little hydrocorals, I think. Very cool yellow encrusting sponge with a bunch of folds. We saw some when we touched the bottom of this feature earlier. I think maybe the arms of a Solaster up to the right, another one of these cookie stars, the Ceramaster in the middle. I was corrected, it was probably eating a sponge, not a coral. Ceramasters seem to prefer sponges. Beautiful. I think could take a peek. It might be hard to make out any features just with the position. That little crab from here looks like it could be something like a decorator crab. I've seen some at similar depths, I think, and I'm seeing the, I don't know, kind of the distinctive banding on the legs. Someone please correct me if I'm wrong. Oh, okay. There's another, yeah, I believe this is a Celaster. Hard to tell without the full view of the center, but looks like it to me. And yeah, I think that looks like a decorator crab. You can see it's covered with little bits of sponge and other debris. Not sure if it got caught up just in this current, but I have the genus Carillia for that little crab.
Now, do they have a way of actually gluing things onto their backs?
I know very little about this one. Just we've spotted it in our video, and they always seem to have, seem to like to maybe disguise themselves. So I don't know if they encourage growth of other organisms or exactly how they do it. That's a pattern. We've seen those urchins earlier on in our survey that like to cover themselves in, dead tubes of things and debris. It seems like a good, I don't know, a popular mode of defense.
Right.
In some of the dives that we've had. ROV watch lead. Can we get a look at that stick? The white stick. I think it's an unbranched Paragorgia. And off to the right, there's a small cloud sponge. We saw a few of them yesterday. The reef-forming sponge, Africalyptes vastus. There we go. So we can see the where the individual polyps are retracted into the tissue, those little bumps. But yeah, this is a small unbranched specimen of one of the bubblegum corals in the genus Paragorgia. There's kind of a species complex of these kind of more sparsely branched, smaller ones. That's great. There's farther up, there's a Primnoa I'd like to look at. See that one sticking out by the sea star? Yep, okay. Sounds good. Wow, look at that. You can just see, pretty much a horizontal movement of the marine snow, and all of the debris in the water column is pretty much raining sideways. Oh, I see that. I think that's a Henricea with a little red spot in the middle. I've seen that. A snap zoom or a zoom would be great, just to know. Yeah, very cool. We haven't seen this this color pattern before, but the rest of it reminds me of a Henricea. Yeah, excellent to document that really interesting color morph. See that anemone closing up on our approach. The pom-pom anemones don't seem to mind us nearly as much.
Little pile of black rocks. I wonder what that's. Are those rocks, or is that?
Yeah, I'd say
Yeah.
that's my guess. A little tiny bit of exposed rock here. Wow. Wow, it is
Sort of a figure-8 worm tube.
Yeah.
Nice.
Yeah, it sure looks like it. More of these little lamp shells or brachiopods. Really curly tube worms. Little egg casing of some kind, and then a bryozoan, I think, or a sponge beside it.
See a little bit more brown on the brachiopods like we were seeing yesterday. So it makes me wonder if those are a different species from the whiter ones, or
And then there's this
just local.
a yellow one over here. These white and brown ones over there. Oh yeah, some really large white ones. So we have confirmed from our science chat that that sea star with the red dot in the middle, confirmed to be a Henricea. I think that genus is a very, very difficult one to differentiate species. So mostly we just leave it at Henricea, especially in our image-based identifications. But even when you have them in the lab, I think they're a pretty difficult group. Oh, that's okay. I'm sure we'll see more. Yep, not worried. Yeah, it feels like we're in a snowstorm. Kind of find myself squinting in the same way. Just the amount of material running through this canal being carried by the current and the tides. Oh, can we look at that sea star straight ahead? It's a little bit darker red in the middle. This might be a target.
Interesting little pile of rocks right there.
Oh, yeah. Looks like that anemone moved them.
It says rock collection.
Looks like it. All right, hopefully Chris Sma can pipe in and confirm, but this looks to me like it might be the sea star he was after yesterday. I've kind of made it my mission today to find one for him. It's an unknown species, potentially of the genus Chiraster. Ooh. So a little bit different. I'm not seeing the banding, but that might not be an identifying feature. I'll wait for Chris to pipe in, but I think this could be the one that we were looking at for our collection. Bear with me for a moment. Let me see. I mean, it does, yeah, it definitely doesn't have the distinct banding of the one in this photo. Yeah, I guess maybe we'll move on just because I'm not sure. Yeah, and I haven't. Oh wait, no, changed. That's it. Just heard from it. That is a collection target. Thank you. Whew, that was close.
That was close, Chris.
I was like, I'm not seeing the banding. I'm not totally sure. But yeah, this is the one. Phew. So NOAA Fisheries puts out a series of poster identification posters that have pictures of all these different sea stars, and it's just down to Chirastor species A. So this collection will hopefully help us fill in that species name. The common name seems to be the Aleutian fragile star.
Really is a very curious pile of boulders.
Yeah, what do you think about that? Like, where do you think they might have come from? Because I don't think they came from up there.
Well, they might be rolling down this little crevice to the right from something. Perhaps a cliffy outcrop.
Yeah, that sounds great. Thanks. Would you want to oh. Whew.
All right.
Can exhale. Wow.
Sample ID EX2306 -D10-03B. Starboard outboard bio box.
Inboard, I think.
Oh, you're right. He did change at the last minute from my guess. Starboard inboard bio box. Thank you.
Mm-hmm. Awesome. Thanks for that. Whew.
ROV watch lead. When we get going, it might be interesting to kind of run up this little V-shaped valley that the boulders are rolling down. If we can, just to see where those are coming from. It's very curious that we've got some boulders that are rolling down here and piling up. Kind of a bowling alley. Maybe nothing up there.
And so these boulders, since you're not sure where they came from, plus they're also a little bit larger, they're not as interesting for like a sample.
Yeah. I'm not sure what anyone would do with them.
Mm-hmm.
We're not sure what unit we're in, and they're actually pretty easily collectible on shore.
Oh, I see. Sure. That makes sense.
So rather than take time to try to pick one up.
Mm-hmm. Some really big individuals of those pinkish anemones we just passed, and I think we're seeing, there's a boot sponge to the left, sedimented on the outside and then kind of clean on the inside. This one is probably Rhabdocalyptus, one of the rosellid sponges.
Yeah, that's fine. That's fine. Yeah, that's good.
Looks like it runs
yeah,
right off the...
I was just curious to see if we'd see what that was coming from, and it might be just coming off this outcrop, but I don't see any obvious thing, but it is a little bit of a steeper face that things might be occasionally falling off of, getting to that little valley V-shaped thing and rolling down to pile up in a single spot.
Mm-hmm. Wow, look at this. It's getting really dramatic. Oh, that helps. It is just like turning off the fog lights when you're driving through snow. So I'm seeing, lots of different encrusting sponges. It's a big solaster down at the bottom. And here's the coral that I'm interested in behind it. If you pan up a little bit, back there in the middle, if we could take a look at this one. Oh, there's a big stick, I think. A lot more woody debris, and it's easy to see why. When we go up on the deck, we're surrounded by these really beautiful scenery. A heavily forested area around where we are.
Well, here's the source of the boulders.
Oh, yeah.
They're all over the place.
You sure you don't want one? They might have cup corals on them.
Well, if there's something that has a cup coral on it, of course.
Okay, here, this is definitely I'd say our Primnoa pacifica. It's got a nice big shrimp hanging out on it, and you can see these really heavily armored polyps have all of these interlocking skeletal elements. They're called sclerites that kind of overlap and almost completely cover the tissue on this coral. Is it possible to sample, get a snip of this? What do you think of the setup here? Okay, I'd like, just a snip. So we sampled a primnoa, I think on one of the seamounts maybe earlier on in this survey, but the area here, there's something very interesting going on with the population genetics, and having this could really fill in a gap. So analyzing the DNA of this coral to give us a better idea of how the populations are distributed throughout the Gulf of Alaska and beyond will be a really interesting question that we can answer with a sample here. A little bit more, maybe, yeah, just a hair, thanks. Yeah, that's good. Yeah. Just a little off the top. Maybe change this. Okay. We'll see this, if that's okay.
Alrighty.
Okay.
EX2306-D10-04B in the starboard outboard bio box.
Excellent. Thanks for that.
And as far as the rocks go, do you see a cup coral on any of those boulders? I actually did kind of have my eye on that really bright, shiny one to the right.
One of these?
Yeah.
Could we do a zoom on those kind of darker colored rocks to the right? It's cup corals. I'll take a ceryanthid. There's a few things that we've been seeing and just been kind of waiting to see the right one. Interesting what the kind of really almost transparent white bushy thing is as well. Oh. Seeing some worm tubes, lots of that brachiopod, and this a really densely branching hydroid, I think, is that white stuff on top, and a dead cup coral. I wouldn't say no to this rock, obviously. There's lots going on if it would be helpful for your work, but otherwise I won't target it, I think. Wow, lots of different kinds of sponges on there, though.
Alrighty. Well, if it doesn't look like a real gem, I'd say let's keep moving and maybe we'll see a better one.
Sure.
But nice to get a look at that.
It's, yeah, very helpful.
Sort of caught my eye with its black face. I was wondering if there wasn't some manganese crust on it, which would have really surprised me. In this environment, as you pan back, it kind of takes on a shiny look that's wrong from...
Yeah, I think we're good. ROV can keep on moving. I'm also keeping an eye out for manganese crust now. I'm starting to train my eye.
Is kind of an interesting rock type. I'm wondering if it's perhaps volcanic, which is something we might expect, at least based on the onshore mapping.
Oh, good. Oh, yeah, for sure. Even just the... Oh, good. Yeah, even just the debris in the water column it's not going sideways anymore, at least.
Visibility isn't a whole lot better, though.
No, we were getting like 1.6s down at the bottom, and now it's more like 1.35.
Copy.
Yeah, some really giant pom-pom anemones, these Liponema. And the other one, I think, had an ID on that one too. Maybe Cribrinopsis. I think some of those little pink branches sticking out should be look like juvenile primnoas to me. You can see there's a rockfish. It's back turned to us, and yeah, a bunch of these like really large anemone bases, partly, you know, kind of the anemones partly retracted into them. Cool. Look at this. Wow. Okay, there's another pretty steep area. Reminds me of what we were looking at yesterday. This is basically seems like pretty much a vertical wall. Little Ceramaster cookie star. Lots of these little, you know, circular things. The darker brown ones are brachiopods. We've seen a couple of different morphotypes. Lots of these anemones, and every now and then some of those little tube-shaped sponges. Lots of encrusting sponges that are kind of growing in a low mat along the face of some of these rocks. It's a very cool one that's that yellow one that's kind of folded in texture. And there's another Primnoa coral up above. And very interesting for us that there are a lot of juveniles, a lot of, you know, small colonies of Primnoa. Oh, there's some more up there. Just coming up on the right. Oh, and a couple of gastropod shells, those big snails or whelks, just beside that big pom-pom anemone. Someone suggested chrysanthemum anemone for that one. I think that's really a really nice one.
Looks like we keep seeing these kind of semi-vertical fractures, reminding me of shear zones, perhaps, or faults. The dominant texture of the rock is these horizontal fractures that may or may not be related to bedding, but they're crisscrossed by those kind of vertical but thinner laminated looking rocks that could be faults.
Ooh,
here we
look
go.
At this. There's a nice big coral that's one of the bubblegum corals, a Paragorgia. So we saw one earlier that was just a little unbranched stick sticking out of a rock, and this one is what it looks like when it is kind of more fully grown. I'd love to get some good imagery of this one. Yeah, yes, please. kind of down in the middle of one of these little ravine almost.
Right.
What did you call it?
A valley? A bowling alley? No.
Yeah, in the bowling alley, yeah. Right on a
This
big
one's got a
boulder in
little
there.
bit more sediment piled up, but yeah, it's making these real interesting, V-shaped features.
It looks like this bubblegum coral is really happy about it. It's a beautiful specimen. I don't know if I've ever seen one. It's quite that regularly branched. It looks like it's oriented, in a fan shape to either take advantage of the current or maybe brace itself in position against, so the current flows a little easier around it. But I'd imagine, usually they kind of fan out into the current so that they can, each of those little individual polyps can grab at the bits of detritus and marine snow that it feeds on. You can see a nice, some kind of brittle star wrapped really tightly around it. So each of those little pink dots against the white tissue is where the polyps are. They're all really tightly retracted right now, probably for a little bit of protection against getting whipped around in this current. But when they're out and feeding, you'll see, the same kinds of polyp form and tentacles of some of the other corals that we've been able to zoom in on. This is a really beautiful one, highly branched. ROV, how would you feel about collecting a piece of this one? Is it too tight in here, or what do you think? Mm-hmm. Sure.
And this valley does, you can see a bunch of those on the right side, a bunch of vertical-looking cracks. That again gives me the impression of a shear zone in through here. Another fault. This is just a real highly deformed rock unit.
Okay. Yeah, maybe either side, just we don't need that much material. So yeah, if we could just get like even a few inches of the tips of one of these. So again, this is another coral where there's a bit of confusion around the taxonomy. So having a piece to examine in detail with the imagery associated with it can really help us. So when we see, these corals in the photo, we can know a little bit more about the species that we're seeing. that can kind of help us to capture what's really going on with the biodiversity in an area. No, definitely not more. Maybe, yeah, like just a little snip from maybe the very sides of one. Just don't want to crush too many branches trying to grab this. Yeah, that would be nice. Yeah, I love that.
Very nice.
Can I change that? Beautiful. Did that go in the outboard? Inboard, thank you.
ID EX2306 underscore D10 underscore 05B in the port inboard bio box.
That's great. Thank you for that collection. Oh, wow.
Oh, look at
all those arrow worms up at the top just floating around by the lights.
They looks like they like the lights,
I
all
think
right.
they do, yeah. You just panned up for a second in the Sirius view and saw, yeah, probably like dozens of little Keatignaths swimming right up by the lights. Yep.
Thanks.
Okay, sounds good. So we're actually coming to the end of this dive. We'll take the last 5 minutes to kind of take a good close look around at this really interesting feature we've found in Behm Canal. It's been very cool to see the abundance of a couple different kinds of corals. I really wasn't expecting to see any bubblegum corals here. So that's a very cool observation and distribution point to have. Also, quite a few juvenile Primnoa pacifica, which could indicate that they're recruiting from an area where there are more abundant, larger colonies of Primnoa close by, or at least they're being brought in from somewhere by the current that we're experiencing here, which has thankfully died down quite a bit since the beginning of our dive. I'm seeing lots of these really beautiful anemones, a couple of really big ones, Liponema, and this pinkish one, possibly Cribronopsis. There's another really, really nice juvenile Primnoa pacifica just at the bottom there. And kind of in the middle, those little pinkish branches. So it's heartening to see. We always like to see coral recruitment in areas where it looks like there isn't any or hasn't been any. Also very excited about that sea star collection that should really help with the identification, narrowing it down to species will be really helpful to know. Oh, there's I think a Hippasteria feeding on one of these little juvenile Primnoas.
Oh, yeah.
Oh, dear. And that red sea cucumber in the middle, that's actually one that we haven't seen a lot of. So I think the little red sea cucumber we saw is in the genus Apostichopus, which is the same genus that has the giant California sea cucumber, if you're familiar with those. Different species, but yeah, they're in the same genus. And still lots and lots of these little brachiopods. They've been all over the place in both dives in the area that we've done so far. So we should be looking at potentially our last dive in this area tomorrow. Another hotspot that is covered in that coral distribution model that I have that I'd really love to ground truth. It looks like it's lighting up as a real hotspot, and we're going to be going right to it. So I am excited to see what it looks like. Some very cool geology for you right at the end here. These little, yeah.
Fractures might be bedding, but it's hard to say.
Oh, cool. There's an egg casing down there that kind of spiral. We zoomed in on
We saw
one.
one of those before, yeah.
We did, yeah. Interesting. Could be from a nudibranch or a gastrobot. They make some really beautiful pattern egg cases.
Watch
yeah,
lead.
watch lead.
Alrighty.
That's great. Sounds good.
Did we want to get a water sample here?
Can we?
Yeah, let's go ahead. Yes.
Yes, thank you. That's
Likely.
right.
We'll let you know.
Beautiful.
That was EX2306-D10-06W, Niskin Bottle 3.
So as we say goodbye to this area of Behm Channel, I'd like to thank everybody, the GFOE, ROV crew, and video crew for persevering on this dive today. I know there was a lot of challenges, and I was really excited to get to see what we saw. 15 minutes, pretty quick ascent.
Then we'll be back in the lab. You've got 3 pretty good bio samples.
Yes,
so,
yeah, no, I'm excited to put us to work a little more than yesterday. I'd also like to make sure I thank the science chat room for all of your input and expertise. It is much appreciated, as always. And I'll preemptively thank everyone that will be helping out in the lab today to sort the samples and make sure they get processed correctly and go to where they need to go. And thanks to NOAA Ocean Exploration and the Okeanos Explorer for making this possible today. So hope to tune in tomorrow from our final dive in this area, and then before we get back out to the deep sea, weather permitting.
Right.
Thanks, everyone.
Nav watch. Yeah, there will be no more Niskins. There is no deep scattering later here, I'm informed. All right. Thank you.